welcome. This is the c.l.s. Education STEM Seminar Series that we've started. For those of you not right here, you're connecting to Canada's only synchrotron research facility, which is situated in the middle-ish of Treaty 6 territory and traditional homeland of the Métis, known as Saskatoon. We would like to acknowledge that most of our education programs are funded at least in part through NSERC Promo Science, and that their flexibility has enabled us to develop ways to build this online and virtual program like this series. And we are very much appreciative of that. If you haven't already met me, my name is Tracy Walker, and I am the Education Programs Lead, working with a spectacular staff, many of whom are here today to help us with this. Anna Maria is with us, Amanda is with us, Jesse is with us, David is kind of connected with us. But before we fully get started, I'd like to just remind a little bit of the logistical bits for this. We are recording so that those unable to join us at this time can still engage later when it suits for them and also with their students. If you prefer to keep your camera off for this reason, feel free to use the chat function if you'd like to engage. There are a couple of different viewing arrangements that you might find helpful, and you can access those through the three little dots on the bottom of your screen to change things around. Gosia will be sharing her screen shortly. I remind you to please keep your microphone on mute unless you're actually trying to talk with us. And we do encourage questions and discussion. If you have a question during Gosia's talk, maybe just put that in the chat and one of us will let her know so that you can pose your question. But there'll be lots of time at the end too to be able to discuss a few things and ask some questions. So yeah, right now I'm excited to introduce to you Dr. Gosia Korbas. She's a senior scientist at the Canadian Light Source and is responsible for the BioXAS-Imaging beamline. In addition to commissioning and running the beamline, she also engages in research and primarily with an interest in mercury and biological tissues. She commits time every year to support student learning in our education programs. She's worked with high school students, undergraduate students, teachers, and teacher candidates. And a lot of those projects have been dealing with chemistry in biological tissues related to environmental issues, which touch on a lot of things that students are interested in and a lot of curriculum, both in biology or health related, but also in chemistry. And then with the beamline, lots of physics too. So there's a lot of curricular connections that can be made. This particular talk was designed to fit with the Saskatchewan Physical Sciences 20 curriculum initially, but there's a lot of different things thrown in. So there's a lot of interesting things to connect. So Gosia, over to you. Thank you so much for joining us. I'm looking forward to this version of your talk.
Thank you, Tracy, and thank you everybody who actually join us right now. I can recognize some names here. Hi, Ward. I didn't say hi before, so I'll start sharing now. Okay. So I think you should be able to see now in the presentation view. So yeah, my talk will be on mercury, and I've been working on mercury for quite some time now and using synchrotron techniques to answer some interesting questions about this very intriguing element. But first, let me start with Canadian Light Source. So this is where I work, and some of you may have been already inside, even as during the public tour. And you may have walked on this mezzanine level and looked inside toward the center. You may have recognized we have this round structures inside. That's our storage ring. That's where we keep our electrons going. And as we bend them with magnets, they produce very bright light. And this pink arrow mark here is actually where the BioXAS beamlines are situated at the storage ring. So underneath we have this white big hatch, and this houses many optical elements for 3 beamlines, because actually we are composed of 3 beamlines. So that's the BioXAS where I work, and conveniently my office is actually just above there. I think it's actually there, or no, actually there. So that's my office. And this is inside that white hatch that you just saw. We have many very shiny boxes in there, and those boxes contain X-ray mirrors. And this one here is actually Monochromator. It basically allows us to tune to a certain X-ray energy that we want to choose from a broad spectrum delivered from the storage ring. BioXAS is dedicated to studies on biological, environmental, and health-related specimens. However, we also do material science as well. We operate with 2 techniques, X-ray absorption spectroscopy. We have 2 beamlines focusing on this technique. And another beamline scientist, Roman Chernikov, who is in charge of those 2 beamlines. And I work on X-ray fluorescence imaging beamline. However, my presentation today will use examples from my previous research, and one of them will actually focus on X-ray absorption spectroscopy, and the other one on X-ray fluorescence imaging. So you will get an overview of how those 2 techniques can help researchers in understanding the complexity of metal interactions in their samples. First, to X-ray absorption spectroscopy. So in very general terms, we use X-ray absorption spectroscopy to determine chemical form of metals, but not only metals, also any other elements that are of interest in research. We call it speciation, so we detect different species of certain element in the samples. What we collect, it's called absorbance spectrum, and this absorbance spectrum is actually shown here, and it will have a very unique shape, so it can be used as a sort of fingerprint of the presence of certain chemical types in the sample. The way we collect the absorbance spectrum is the following. So we have X-rays shining on the sample and passing through the sample, and then we in a very simple term, of course, it can get complicated, but in a transmission mode, as we call it, we detect and measure intensity of the beam before the sample and intensity of the beam after the sample. We use those 2 values to calculate absorbance, and then we plot absorbance versus X-ray energy. So we measure this absorbance for a series of different energies, and each energy is actually chosen from a broad spectrum through this monochromator that I was showing you before. What we can stick in the beam is actually really up to us. It can be soil that was actually mentioned, could be metalloproteins are quite often used, but definitely intact biological tissues as well. Can be material science samples. The other technique is X-ray fluorescence imaging. So this is for available on BioXAS-Imaging beamline that just recently got commissioned and will be opening to the general users, as we call it. This technique will not provide spectra to the users, will provide images. And these are images of distribution of metals and other elements of interest to the researchers. We can measure various samples as well. Quite often we measure sections of organs or tissues. We can measure whole plants, cell cultures as well. And depending on the equipment that is available on the beamline and the beam spot sizes, which determine resolution, and I will touch on that later in my presentation, you can also measure quite big objects like fossils and paintings. Most often the beam that comes from the storage ring is too big for this sort of experiments, so we have to make it smaller first. And we use various optical elements to just make it as thin as few microns or even less. And then with this small X-ray microbeam, we raster scan our sample in this beam. So beam stays stable, we are moving the sample in the beam, and spot by spot, we are irradiating the sample and collect X-ray spectra with X-ray detector here. And then we analyze the spectra, and what comes out, it's basically a map of intensities corresponding to different elements in the same sample. So from one measurement, you will get multiple images. You can get an image of sulfur shown here, or calcium or mercury, basically whatever you are interested and whatever the beamline was set up to measure. Those two techniques are very important in studying elements, and especially elements that have impact on our health or wildlife health. We can divide them as beneficial or toxic, though sometimes this differentiation may be quite complicated, and I will mention this in a moment. Beneficial elements are, for example, cobalt, iron, zinc, copper, molybdenum, manganese, selenium. Toxic ones, I'm sure that anybody can mention some. Arsenic, it's obvious choice, cadmium, beryllium, lead for sure, mercury, the one that I will be talking about, thallium, selenium. Selenium, as you may have noticed, I actually have in this group and in the toxic group as well. And yes, this is one of the elements that can actually be both, depending on the speciation of it and concentrations. It can be actually either beneficial or toxic. And concentration is, of course, very important. This was first determined by actually Paracelsus. He was a Swiss medical doctor back in the 16th century. He was considered a free thinker and actually a founder of modern toxicology. And he was the one who says the first time that solely the dose determines that the thing is not a poison. And this is definitely true. It can be also true for beneficial elements. So now to my favorite element, mercury. This is a naturally occurring element in the Earth crust in the metallic form. So you may remember that form from thermometers if you are old enough. We can release mercury to atmosphere through natural sources like volcano eruptions, but we can also release mercury through coal burning and small-scale gold mining. I will not have time to touch on small-scale gold mining, but if there are any teachers tuning in, I would encourage you to look into it and to educate children in the classroom on small-scale gold mining. Because unfortunately, lots of small kids in many countries around the globe are involved in the process of this gold mining, and this gold mining involves mercury, unfortunately, and actually burning elemental mercury, which releases vapors and poisons the miners. So it's a very important topic these days. As a small interesting things about mercury, which I found interesting, and I think the more you dig about mercury, you will find more of those from the past. So Paracelsus, as I mentioned before, he actually formulated the first ointment that contained mercury for the treatment of syphilis. You may recognize the term Mad as a Hatter or Mad Hatter from Alice in the Wonderland. And this actually stems from 1800s, where people were using mercurous nitride for making felt hats. And by inhaling the toxic vapors, they were actually going mad. And that's why we have this term now, Mad as a Hatter. Mercury, unfortunately, is still used in everyday products. I mentioned thermometers, though I think it's less widely available these days. But also some switches, thermostats, batteries, fluorescence lamps for sure, and in some traditional medicines and skin lightening creams. You may have heard about the story, it was not so long ago on the news, about the woman in California who actually ordered a skin cream from Mexico, and unfortunately that skin cream was tainted with organic mercury. So it's still happening around the globe. Fortunately, though, since 2017, we have what we call Minamata Convention on Mercury. It's a global treaty that has been signed by many countries, and I think there are still countries signing it these days. And that was built to protect human health, but also the environment from the toxic effects of mercury, and to phase out mercury products altogether. Mercury chemistry is really important, and especially in biological tissues, because it sort of determines what really happens to mercury. We have elemental mercury. Again, you may remember from thermometers. We have inorganic mercury and organic mercury. In organic mercury, we have mercury atom covalently bound to a carbon in this case, this is a methyl group, but it could be any alkyl group actually. And what's really the most important thing when it comes to mercury chemistry is that both inorganic and organic form will have a very strong affinity towards sulfur and selenium, and specifically to the form of thiol. So you can actually see here an alkyl group bound to SH, or selenol, so alkyl group bound to SEH. Why speciation matters for mercury? I mean, why? I think it's a very good question, but it definitely matters. So I have an example of two compounds. So one is methyl mercury cysteínate, and the other one is alpha mercuric sulfide called cinnabar. Both of them contain mercury. One is inorganic, one is organic. However, they significantly differ in their toxicity and generally toxicological profiles. Methyl mercury cysteínate is present in fish and seafood products. And what really makes it so toxic, first of all, is that it's readily bioavailable. So when you ingest fish, more than 95% of that form from the fish meat will actually be absorbed in the gastrointestinal tract and will get into your bloodstream. Once it's there, there is another issue. The endothelial cells that are lining our blood vessels in the brain, their main purpose is to protect the brain from harmful chemicals. However, methyl mercury cysteínate is quite sneaky and pretends to be something else, and this way it gets into the brain. So it will accumulate in the central nervous system, and the same situation, or the same scenario also happens when it comes to blood placental barrier. So it will also accumulate in developing fetuses. It is therefore considered a very potent neurotoxin. In contrast, we have a cinnabar. It's a naturally occurring ore. It has been used in traditional Chinese medicine. It has a beautiful bright red color. That's why it was also used as a red pigment in tattoo dyes. As you can see here, only 0.2% is absorbed from the gastrointestinal tract. And also, therefore, one of the reasons for that is that its neurotoxic potency will be considered 1,000 times less than of methyl mercury. I touched on that a little bit earlier. So because methyl mercury accumulates in the central nervous system, it will cause some toxic effects on the brain, and especially on the developing brain. So people poisoned with methyl mercury will complain on paresthesia, ataxia. They will experience sensory disturbances, tremors, vision and hearing problems. They will have difficulty walking. And especially I would like to bring your attention to the sensory problems, so vision and hearing problems, because what we found out shed some light on those impairments. Risks are the greatest for infants and unborn children, and methyl mercury likely interferes with several critical processes that are happening during the development of the fetuses that are actually mentioned here. And these days, there is more and more evidence that even lower exposures are contributing to heart diseases and cognitive deficits, not only in children. So the main problem really now with methyl mercury is that I haven't mentioned something. We have something called mercury global site. So all this inorganic mercury that is emitted to the atmosphere, either from natural or human human-made sources, it's 80% of it will come back to the ocean. So it will be deposited in the ocean as inorganic mercury. However, there are microorganisms in the ocean that convert this inorganic mercury into metal mercury. Predatory fish will then amplify those metal mercury concentrations by million times of more. And around the globe, we have more than 3 billion people relying on seafood for nutrition. So because fish are the main source of nutrition for at least those 3 billion people, we have a problem with human exposure to metal mercury. And to make things worse, recent studies are also showing that climate change and overfishing is contributing to and will contribute in the future to increased levels of metal mercury in marine predators. And just for an illustration, you have a link here if you would be interested. That's not my research, but I found a really good illustration of concerns that are expressed these days in mercury community. You can see effect of seawater warming in the Gulf of Maine on tissue metal mercury in this Atlantic bluefin tuna. And in the gray zone, we have projections of how much they are expecting it will increase and how much it's already starting to increase. And these are the levels, relative levels of metal mercury in the bluefin tuna tissues. So we are talking, due to seawater warming, we most likely will see tens of % increase in the metal mercury concentration. I need to also mention something that actually Minamata name came as Minamata Convention, but actually it comes from a long time ago when there was a huge Minamata outbreak. It happened in Japan in the 60s, and actually it started in 1956. So that was a small bay where there were lots of, it was basically a fishing village. And next to it, there was a Shisho Minamata factory that was producing acetaldehyde, and they were using mercury sulfide as a catalyst, and side reaction led to the production of metal mercury. All the wastewater was then dumped into the Minamata Bay and contaminated the fish there. The first official report, as I mentioned, was released in 1956, and there were typical symptoms of mercury poisoning, but they were not recognized for some time. There was also some cover-up by the government, of course, by the factory. The interesting part was also that not only humans were getting sick, but also pets and other animals. So there are some really striking reports of pets experiencing convulsions, going mad, dying, birds falling from the sky, fish floating belly up in the bay. So it was pretty extreme what was going on there. Finally, it was recognized as a mercury poisoning. The maximum mercury level in hair detected was more than 700 ppm. So you can see how high it is, because normal levels you wouldn't expect more than 1 ppm. This pollution continued until 1965, but this Minamata outbreak brought global community, attention of global community to mercury problem, and especially problem in developing fetuses, because there were reports of mothers that were not getting sick, but giving birth to children significantly impaired, either mentally or physically. There is a Canadian connection here. So you may have heard about Grassy Narrows. It's a First Nation community in the north of Ontario. And then there was a Dryden Chemicals company that used mercury in the process of bleaching paper. And again, the same sort of story. They were dumping this mercury contaminated waste into the English Wabigoon River. This hasn't been really, population were complaining about, symptoms of mercury poisoning, but has this for a long time continued and was not really taken seriously. 2018, Ontario government finally pledged 85 million towards cleanup and also construction of a care center. In 2019, Supreme Court ruled companies responsible to pay for that cleanup. And very recently, actually in April 2020, federal government actually signed agreements to fund that, the cost of the building, this Mercury care home, as the community is calling it. for the members suffering from mercury poisoning. So yeah, we have a very local connection in Canada as well. So now to the examples. So this example will actually illustrate X-ray fluorescence imaging technique. And we use this technique to determine how different absorption of inorganic and organic mercury is in the, especially in developing vertebrates. We use zebrafish as a model system. It's a very nice model system to work with. We didn't use adult zebrafish. So zebrafish is actually the same fish that you can get in pet store. They are used in many studies in developmental biology, and also as a model system to study human diseases. What's really useful when it comes to zebrafish is, first of all, that for the first few days of their life, they are fully transparent. And then for the 7 days, up to 7 days, they still feed from their yolk. So when you have them in water, like you can see here, a petri dish full of fertilized eggs, they actually don't require any external feeding. So you can just spike the water with whatever compound you are looking at. And this way you can expose them to these harmful chemicals. Imagine if you were to add some food. Food can also bind this, let's say mercury, for example, and interfere with the absorbance, or it can even change the chemical form. So it's a very good model to work with, and especially these days it is used, as I said, to study human diseases, because believe it or not, but 70% of protein coding human genes, they actually have their counterparts in zebrafish, and also 84% of genes that are linked to human diseases can be found in zebrafish. So this is really the main result that we got. We exposed fish to 1 micromolar either mercury chloride or methyl mercury chloride here. So the amount of mercury in water was the same. We exposed very young fish. I think they were around 3 days post-fertilization. So more or less at that stage, they looked like this. And we looked at 3 different areas. We first checked a section through their nose, a section through their head, including the eye. And this was the section through their gastrointestinal tract. On the top panel here, the blue-looking images are histological sections through those areas. They are only 6 micron thick, and they are blue because they were actually stained with a dye called methylene blue to reveal all the different cells. Otherwise, the tissue looks very blunt. It's almost transparent, and it would be really hard for us to do the comparison. These are the images from the synchrotron. They show mercury distribution in the adjacent section. So we always take one which we stain with a dye, and then the second one comes for the synchrotron and is not treated in any way. And then we measure mercury distribution together with distribution of other elements in those tissues. Also, the thickness of this tissue is also 6 microns. We use 5 micron beam for that specific experiment. And then when we plotted mercury distribution for each organ, we plotted them on the same scale. So blue, it means almost nothing. And then, of course, the maximum level in either of those images would be correct. So the main result you can already see here, that when we look at the head section from a zebrafish larva that was exposed to inorganic mercury, we barely could see any mercury accumulating. When it came to methyl mercury exposure, we definitely saw mercury accumulating in the brain and also in the eye. This was really the main interesting result, because it has never really been reported before, that accumulation of mercury in the lens. So this is the eye lens in the fish. And those different layers that you can see here, these are the cellular layers in the retina. So in addition to the lens, we also saw that layer of mercury around the retina. And at that time, at that resolution that we had, we were attributing this mercury to the accumulation in this, what it's called, retinal pigmented epithelium. But then we verified actually our results with much smaller beam. The same sort of result was also observed in the fish that was treated with organic mercury in the gastrointestinal tract. So what you are seeing here, actually this area here and there, this is the liver. Then you have basically this is the stomach of the fish, the gut, the lining of the fish. This is big yolk, so this is where all the nutrients are, part of the brain and muscles. So organic mercury definitely accumulated at much higher levels in the fish than organic mercury. And interestingly, only the same levels were seen in the nose. So what you can see here is the lining of the nose of fish, and both forms of mercury, organic here and inorganic here, at the same levels, they accumulated in those epithelial cells. So for us, really, the main take from that was accumulation, specific accumulation of organic mercury in the eye, in one of the main sensory organs. But we also saw accumulation in other sensory organs. So previous images were taken at 5-micron resolution, and then we were really interested in this retina, exactly where we can see mercury, and also in the lens, because it was really hard to say at 5 microns where exactly. So this is a nice illustration what significantly smaller beam can do for you. So if you have 5-micron beam, these are the images that you can get, shown on the previous slide. This was the beam at 1/4 of a micron. And this definitely allowed us to resolve between different layers in the lens. So you have a histological image here, and the rectangular area that was imaged at the synchrotron. This is still just zoom in into that portion here. You can see CR marks cornea, EP marks epithelial cells. So developing lens will always have this single cellular layer of epithelial cells that multiply, and then once they become older, then differentiate into lens fibers. And with that resolution that we had, we could definitely, and by comparing, for example, with images of calcium distribution and overlaying those elemental distributions, so mercury would be blue here, sulfur red, calcium green. You can see that calcium marks nicely cornea and epithelial cells. And then sulfur and mercury colocalized in this area where the lens fibers were. So definitely not actually where we initially expected, because we actually expected mercury to be more accumulating in that most peripheral area of the lens. The most interesting result came though from the retina. So as I said, we were speculating that it would be actually in this black pigment here, but it turned out that actually it wasn't. The story which was much more interesting because pigment is known to accumulate metals in general. I think not just in fish, in humans as well. So that wouldn't be that novel, but actually when we image at the same 1/4 of a micron resolution section through the retina, shown actually on that histological image, and again we plotted together phosphorus this time, zinc and mercury, we could definitely see accumulation of mercury blue. You can see here actually there, and what you are looking at, these are photoreceptors. So then we tried even smaller resolution at 100 nanometers, sorry. So just to focus on those photoreceptors. And then comparing what we are seeing. So these were the images that were, sorry, I have to bring something down because I cannot really see my full screen. Okay, so you can see that we measure phosphorus, sulfur, zinc, mercury, the same as before. Phosphorus is really good signal because it will show you where cell nuclei are. Sulfur always colocalizes with mercury. This is a nice illustration, what I mentioned before, of this thermodynamic affinity. And zinc is one of the elements that it's often detected in pigments. So zinc marks nicely this pigmented epithelium, and then we have phosphorus marking cell nuclei of the photoreceptors. And then mercury mostly was actually detected above those cell nuclei. So you can see phosphorus signal here, and then mercury blue in the above portion of the photoreceptor. So when you look at the structure of photoreceptors, we have either rods or cones. We didn't have enough resolution to actually determine at which ones are mostly accumulating metal mercury. So that's also something interesting to look into later. But generally, photoreceptors actually build of this portion when you have a cell nucleus here, and then above you have stacks of membranes that contain a molecule called rhodopsin, and rhodopsin is involved in interacting directly with light. And rhodopsin will also contain lots of thiol groups, so sulfur. So most likely this is why mercury is actually binding to those stacks of membranes. So this is, you can start from really big, small, relatively not that good resolution, and then it can really, you can bring it down to this, nano levels, and you can start seeing more and more. So really the main things that we learned from zebrafish was that definitely islands and the retina were specifically targeted by organic mercury. That metal mercury accumulated specifically in the retinal photoreceptors and in the outer segments of the photoreceptors. And really the main result was that up to this point, all these visual problems that people experience due to mercury poisoning were attributed to accumulation of metal mercury in the brain, and especially in the area called visual cortex, where all the visual stimuli are processed. But thanks to our research, we could also say that it's not necessarily just in the brain, it's also directly in the eye and in the photoreceptor. So it is likely that this accumulation causes all the disturbances that people experience. And another example, that this example will nicely illustrate how effective X-ray absorption spectroscopy technique is in speciation of elements of interest. In this case, it was mercury. That's really a main question, I think, in mercury toxicology, like what exactly happens with all this metal mercury that you consume with fish meat. Or, I mean, that happens, of course, way less often, if somebody accidentally gets poisoned with high levels of metal mercury. What really happens in the brain? Because this is where the most damage is done. We had access to very unique brains. So we call them case 1, case 2. And both, they came from females. One female was poisoned with dimethyl mercury. It was through the skin contact in a research laboratory. Dimethyl mercury is also an organic mercury, likely the most toxic form of organic mercury. You cannot really get, it's produced a little bit in the ocean actually, but it's very tiny levels, minuscule that you wouldn't even care about. But this dimethyl mercury, it's very poisonous for humans. So she actually died 10 months following the exposure. And case 2 was also female. She actually was poisoned through consumption of a contaminated pork with organic mercury. That happened when she was 8 years old, but she survived for 21 years. So really it was a very interesting comparison, because we have very two different time frames between time spans between the exposure and the death. Again, we collected the absorbance spectrum. The samples were actually coming from the brain cortex of those individuals. And then we tried to reproduce the shape of this X-ray absorption spectrum with absorbance spectra of the model compounds. We considered, of course, multiple model compounds, but in the end, we were able to reproduce this shape. So this is your experimental spectrum in black, and then the green, light green line shows you the linear combination. So in this case, for case 1, we were able to reproduce this shape with 55% of methyl mercury cystainate, so shown actually here as a molecule, 29% of mercury selenite, but in the form of nanoparticles, because the shape of the X-ray absorption spectrum is different for the bulk mineral and for the nanoparticles. And 16% remaining portion was mercury sulfide. Sorry, it was something close to basically mercury bis-cystainate, something along those lines. So it was, we cannot really say what this R group would be. And then when we plotted together, I think this nicely illustrates what really happens or what we think might happen. So when there was only 10 months between the exposure and the death, we could still see the original form that was accumulating in the brain, so organic form, this methyl mercury cystainate, and a little bit of other inorganic forms. And that portion was, of course, very interesting because it contained selenium. And selenium, as I said, is a beneficial element. It's a very important element for the brain function. So that was definitely something interesting, especially that it was in the form of nanoparticles. However, look what happens when we are 21 years post-exposure. The original form was completely gone. We couldn't really see any organic mercury anymore, and we only could detect the other two inorganic forms. And especially this nanoparticles mercury selenite was dominating form in this brain. So this told us a little bit of a story, but I think there is still more to it. So it does look like that methyl mercury eventually will get transformed into this mercury selenite in the form of nanoparticles. But we have to remember that toxicity of bulk material and nanoparticles is significantly different. And it's not just true for mercury selenite, it's true for any minerals, actually. So it's really hard to say at this moment what exactly happens. But I think organic mercury is transformed eventually, maybe through some intermediate compounds into this mercury selenite in the forms of tiny particles. But we still don't know what the source of selenium is, because I didn't really show the results here, but another portion of that research was also looking at speciation of selenium and quantifying the selenium in the brain samples, and we couldn't really see any changes in selenium, organic selenium, which is the form that is present in the brain. So it's really hard to say at this moment from where that selenium is coming from, but it definitely sheds some light on something what is called selenium depletion hypothesis. So basically this one, it's still a hypothesis which claims that all these detrimental effects of mercury accumulation in the brain are actually coming because we are depleting selenium from the brain. And you can actually see here, we have a mercury selenite form in the brain. That selenium must be coming from somewhere. And if it's coming from somewhere, it's missing somewhere else, which is likely not very good. And with this sort of open question, I will end my presentation. And I thank you all for the attention. And for all the people listening, but also maybe listening later on, you can find all the contact info here. And I love answering questions. I love working with students. So yeah, I'm definitely available if you would like to get in touch. Thank you.
Well, thank you very much. That was really informative. And there's lots of connections to curriculum that I can think of. So you have worked with a number of high school groups in their research projects, and none of those groups were looking at mercury in biological systems, but they were looking at chemistry and metals in systems. Which of those groups was sort of your favorite project?
Oh, well, I think I will always have fond memories, of, so maybe, just a little bit of background, I think, for people that, that are not from c.l.s. Yeah. So I started working at c.l.s. when there was no BioXAS. We were building BioXAS. So when I was working with high school students group, we would use different beamlines. But I will always, I think, have fond memories, of two groups. So one group that was the first group that I accommodated on BioXAS site. So we just started doing some experiments, and they were looking at some issues. But if probably the most, I will still remember the first group. It was a group from BC, and they were looking at seaweed samples and copper accumulation. And it was such an interesting project because the kids actually collected their own samples. They exposed them to copper. They were very engaged. They had aquarium at their home actually because it required quite a lot of space. And it time that we run a very successful imaging experiment on BioXAS-Imaging together with EB. So not really, we haven't run that many, but I think that was the first group that we really got some nice looking images of not only copper but many other element distribution in seaweed samples. So I think from that perspective, I guess I enjoyed most, but I really loved working with each of them, and I was even looking for a picture yesterday to sort of finish my presentation. And so I went through like some of the groups. And I mean, each of the project was very interesting. I'm always impressed, that those kids, they put so much work into it and they are so interested. And I hope that they just, it's sort of experience of lifetime. It always sounds like it, when they present. So I enjoy working with students.
Thank you for that. I was hoping that you would share a little bit about one of those experiments, and you did. Thank you. For the teachers who are watching this to understand that if they're going to work with students, they're unlikely going to be able to look at the brains of humans.
Yes.
They may or may not be able to look at biological tissues of what was at one point a living sample, because it's not impossible. We had a student group that looked at mussels not long ago with you.
Mm-hmm. Yeah.
So it is possible.
Project. Yeah.
But we can also do things with plants like the seaweed or other plants and things like that, which are a little more amenable to connecting a classroom with or students during a class.
Yeah, for sure. And for if it was a project for BioXAS, then we can measure small things with small beam, but we can also measure quite big things. Big things, as size of a human brain or bigger, but obviously not human brain. So it could be quite a big plant actually that we could image as well. Yeah.
Are there questions from others?
I have a question. Hey, Gosia, I loved your presentation. I can listen to your presentations all the time. But I haven't seen, when you compare now these two projects, the zebrafish and then bringing and covering the human brain and human samples. So has been any study done on human eye before, or just your zebrafish that you have done on the eye?
Yeah, regarding the eye, from what I remember, because that's the project I need to of course mention and acknowledge. That was all done together with Doctors Graham, George, and Pickering. This is where from University of Saskatchewan. This is where I was a postdoc and then a research associate.
Yeah.
So I remember we were really interested, and I think Graham at some point dig out a very old paper. It was not even, I think, a paper. It was like a book, collection of papers on, I think it was rats or mice. And this was done with methyl mercury that was this radioactive one. I think it's 203 mercury. And they actually had some cool results. It did look like it was accumulated in the lens. This was, I think, study, people did some crazy research, interesting, crazy research back in the 60s or 70s. So I think it was around this time. But I think it was really the only one that fine. I mean, there are some reports, I think, of some heavy metal accumulation in the lens. But I think it was more in the context of some other metals, not mercury per se.
Because I'm asking because you mentioned that the zebrafish were actually fed in the water, like spiked in the water. So isn't it that not necessary the organism needs to ingest the methyl mercury in order to accumulate it in the lens? It could go through directly from the water, or
it could, and when we were trying to publish the story, I remember that was always the main issue. If this is, maybe it's just the diffusion. It diffused the cornea and then got into the eye. But in this paper, we actually did some, calculations, some modeling, and it could not really be attributed to a diffusion, and especially that we have this very specific accumulation. It may be though, so what I'm trying to say, that is probably, it was more like an active transport. But it's possible, that when fish is actually ingesting food that contaminated with methyl mercury, this accumulation may look different. It may not necessarily be in the same area, and that's also, would be very interesting to see. We also did not expose adults, so we don't know what's happening, with adults. We really just work with the young larvae, and it may also be that, maybe they have some more leaky system still at that age. Though I think we definitely looked at, I didn't mention, but in addition to like blood placental and blood-brain barrier, there's also blood-retinal barrier. And it actually is present. It's supposed to be already developed at 3 days post-fertilization, but it may be still quite leaky. So it's there are many interesting questions out there. Yeah, definitely, it's a hint that there may be something happening, but is it happening all the time in humans, including humans? I don't know.
Okay, thank you.
Other questions? Quiet group that we have here today.
It's not always quantity, quality also counts, These are very good questions that we got. So I think, and yeah, I'm very grateful for all the, positive feedback I heard today. Thank you.
To say thank you. It's the first time I've actually learned about the Synchrotron, so it's nice to know more of what you're doing there.
Yeah, no, for sure. I mean, if it was not for this difficult times that we live in, I would say just come and visit us at c.l.s. Unfortunately, that's not that easy at this moment, but I mean, anytime. As I mentioned, I'm always happy to answer questions, or if people have projects in mind, possibly using BioXAS as well for the students. I will be happy to answer any questions you may have. And it's both me and Evie that you heard, who is an associate scientist that works with me on BioXAS-Imaging. So we are both usually working with students group, and I mean, we both would be happy to be involved.
So Gosia and also Evie, and maybe I'll pick on Ward too, and David, since these are all c.l.s. staff folks that are on this call. If teachers have questions or they'd like to sort of discuss some aspect that is not directly related to doing a project, maybe, but some piece of their curriculum that they would like to engage with someone to explain more fully. So maybe it is about X-ray fluorescence, or maybe it's about the retina of an eye and how that works out, just to connect to your talk, Gosia. Would you be willing to have a conversation, like a virtual meeting like this, with teachers and or students about a specific topic?
Oh yeah, no, me for sure. You know me, Tracy. You don't need to ask, always.
Yeah, absolutely. The education team has a really good idea of what the strengths of the various staff at the c.l.s. are, so I have no doubt that they can track down the appropriate person to have a conversation with.
Yeah,
it's
sure.
happened before, and it will again. It's an excellent opportunity.
Sure thing, Tracy.
I take it you can all see the headlights of the bus that's about to run you over?
It's just another day at the office.
Sorry, I've got somebody's mowing the lawn next door. So when I do end up finding out what I'm teaching, I will definitely see if I can add in some content with you guys. That would be awesome. Because I don't even know. I've been told you might be teaching some sciences, but we don't know yet. So yeah, it's been fun.
There's a lot that's still up in the air, and I'm giggling because I know some of the story behind behind her comments. So those of you on the call that are teachers, or who may be watching this afterwards who are teachers, that saw some small tidbit in Gosia's talk, or if you see anybody else's talk in this, and there's something that you would like to reconnect about, and expand on a smaller piece of it, that's something that we're certainly open to. If you get a hold of anyone on the education team, we can set that up for you. Okay. With that, thank you very much, Gosia, for your time and your,
thank you.
your stories. It's really good. It's always a pleasure. Thank you so much.
Thank you all for coming.